Namaskar, welcome to PK's classes. Today we will study anti-cancer antibiotics. As you know, antibiotics are the metabolic products of microorganisms, usually bacteria and fungus, used to kill or inhibit the growth of other microorganisms like bacteria, fungus. So they can be antibacterial, antifungal, and they can also be used against cancer cells. So they can also be used as anti-cancer. So antibiotics, some of the metabolic products of microorganisms, that is the antibiotics, can also have anti-cancer use. So they are called as anti-cancer antibiotics or antineoplastic antibiotics. So as you know, the anti-cancer drugs can be cytotoxic drugs, hormonal drugs, and targeted drugs. And among the cytotoxic drugs, one group is antibiotics. So as per the definition, only those drugs whose source is microorganisms, they are used against cancer, then only they are called as antineoplastic or anti-cancer antibiotics. And there can be four groups of anti-cancer antibiotics. Anthracyclines, for example, doxorubicin, daunorubicin, epirubicin, idarubicin. Then phenoxyazine antibiotics, actinomycin D or dactinomycin. Then aziridine antibiotics, aziridine and quinone are present. So mitomycin C, they act as alkylating agents. Then glycopeptide antibiotics, for example, bleomycin. So let us discuss them one by one. So one of the first antibiotic against cancer is actinomycin D or dactinomycin, and the chromophore is this phenoxyazine ring, which is a tricyclic ring. So they can be called as phenoxyazine antibiotics. And the source is Streptomyces antibioticus. So it is an antibiotic because it is derived from the bacteria Streptomyces. So this antibiotic, you can say it is the first antibiotic to find therapeutic application in cancer chemotherapy, actinomycin D. And if we discuss the mechanism of action, it causes intercalation of DNA, first one, and it also inhibits RNA synthesis by inhibiting DNA dependent RNA polymerase. So it inhibits mainly messenger RNA synthesis. And these are the two main mechanisms of action, and in addition it can also induce DNA strand break by inhibiting topoisomerase 2 by generation of free radicals. Also another mechanism can be there, by it can cause DNA strand breaks by generation of free radicals. But the major one is intercalation of DNA and inhibition of mRNA synthesis. And actinomycin D can be used in Wilms tumor or nephroblastoma, kidney cancer, Ewing's sarcoma, which is related to your bone in children and young adults, then rhabdomyosarcoma related to muscles. And they show toxicity like bone marrow suppression, mucositis. Next we will go to the second group of anti-cancer antibiotics, that is anthracyclines, and they were first isolated from Streptomyces pusetius. So again the bacteria from which they are isolated, and it was developed in 1958, this group of antibiotics, and their major mechanism is DNA intercalation and inhibition of topoisomerase, and they are most effective cancer drugs with a very wide spectrum, can be used against breast cancer, leukemia, lymphoma, sarcomas. So these 4 antibiotics, doxorubicin, daunorubicin, idarubicin, epirubicin, these are all coming under anthracycline class of anti-cancer antibiotics. So the mechanism of action in detail: one is intercalation of base pairs, inhibit topoisomerase 2, and generate free radicals like superoxide, hydrogen peroxide, which cause breaks in the DNA, and in addition they also inhibit the RNA and DNA synthesis. So one is DNA strand breaks through intercalation of DNA and inhibition of topoisomerase 2. The free radicals, the reactive oxygen species, can also cause cell damage, ultimately lead to cell death. So they have the toxicity, cardiotoxicity, which is dose dependent. Then the third group of anti-cancer antibiotics containing quinone ring and the aziridine group, and example is mitomycin C, and it is isolated from Streptomyces cyspitosus. So this is antibiotic because it is derived from this bacteria. It is, if you discuss the mechanism of action, it acts as an alkylating agent, and it alkylates the DNA, thereby causing breakage in the DNA strand and hence inhibition of the DNA synthesis. And it is used against gastric cancer, pancreatic cancer, bladder cancer, and in combination with vincristine, the vinca alkaloid can be used against breast cancer. Adverse effects: myelosuppression, pulmonary toxicity, that is pulmonary fibrosis, nephrotoxicity. These are the toxicities associated with mitomycin C. Then finally we have the glycopeptide antibiotic, that is bleomycin, and it is called antibiotic because it is derived from the bacterium Streptomyces verticillus, and it refers to a family of structurally related compounds, basically bleomycin A2 and bleomycin B2. And it mechanism of action, it causes fragmentation of DNA, breakage of DNA. So it generates free radicals which causes DNA fragmentation. So DNA, bleomycin, and iron complex appears to undergo oxidation to bleomycin ferric complex. So DNA bleomycin ferrous undergoes oxidation to bleomycin ferric complex and liberates electron, which react with the oxygen to form the superoxide, and then further generates the hydroxyl radical, and they cause the fragmentation or breakage of DNA. They can be used against testicular cancer, so it has the ability to cure testicular cancer, and can be used against Hodgkin's lymphoma, though it is not curative. Then can also be used in squamous cell carcinoma. In addition, it can be used in the treatment of plantar warts. And toxicities associated: pulmonary toxicity, skin toxicity, and bone marrow suppression, but it is minimal as compared to other antibiotics. So pulmonary fibrosis, skin rash, and bone marrow suppression. These are the toxicity associated with bleomycin. So that's all about the anti-cancer or antineoplastic antibiotics. Thank you.